Hello, my name is Lisa Rudd, and today I'm going to be discussing my project for Tools of Biochemistry, the technique of ELISA. The paper that I'm going to be reviewing is called A Multiplex Immunoassay for the Non-Invasive Detection of Bladder Cancer, which was published in the Journal of Translational Medicine in 2016. So this topic relates to Chapter 7, which we went over in our book. One section of Chapter 7 discusses antibodies, which are a major group of proteins involved in our immune system. And what is unique about them is that they have a great deal of amino acid variation, and this results in a high level of specificity to unique cellular targets. So antibodies are going to be our best defense against foreign invaders such as bacteria and viral pathogens. Antibodies are involved in recognition of self and non-self, and in the lab, antibodies can be made against biological targets because of this high level of specificity. IgG is most commonly used type of antibody for biochemical and biotech applications. It is a Y-shaped, contains Y-shaped monomers with two antigen binding sites, and we have antibody-antigen reactions that are involved at those sites that can be detected with techniques such as ELISA. ELISA can be run in three different formats: direct, indirect, and sandwich. And today I'm going to be talking about the sandwich format with my talk. So with the IgG, as we learned in the book, that we have, a heavy chain region at the base. We also have a hinge region and a light chain region. And then at the ends of the Ys, we have the antigen binding sites. And these are important for specificity of our technique and in our detection. So, with the 3D structure, this also shows what this Y-shaped IgG looks like. And again, at the tips we have our antigen binding sites, and when you have amino acid variations with your antibody, that confers specificity. So our technique today is going to involve sandwich ELISA. It's a very highly sensitive technique. It involves quantitating antigens between that are bound between two layers of antibodies. And basically, when you use this technique, you can use either monoclonal or polyclonal antibodies and for your capture and detection. So, you have that choice there, but it is very highly sensitive, and it's actually 2 to 3 times more sensitive than your direct or your indirect ELISA, because of having your sandwich layer of antibodies and that amount of specificity. So our technique for ELISA is going to involve having basically a 96 well plate format, and you're going to have a plate that's going to be coated with a capture antibody, and that's illustrated in 1. Then you add your sample, and any antigen that would be present in that sample is going to bind to that capture antibody. Then the second part of the sandwich would be your detector antibody will bind to your antigen. And so then in diagram 4, you have an enzyme linked secondary antibody that will be used for your determining and detecting your, for your detection technique. So, and then by itself, that output cannot be read, so you have to add a substrate that will convert by enzyme into a form that's going to be detectable, and that can either be based on change in color, change in fluorescence, or change in electrochemical signal. And this is usually read in a microplate reader. So here are the steps listed out a little bit long in a little bit more detailed format. So again, you prepare your surface. You could block any specific binding on the site. You apply your sample that contains your antigen, and then you wash your plate in step 4, so that way anything that's unbound is going to be removed. Then you add your specific antibody, which will bind to your antigen. You apply your enzyme-linked secondary antibody as your detector antibody. You wash a plate to remove any unbound antibody enzyme conjugates, and then you apply a chemical that will convert it to a form for detection. So, summary of the technique. A sandwich ELISA is going to have high level specificity because you have 2 antibodies involved in your antigen antibody pair for capture and detection. So it's going to be suitable for complex samples since your antigen does not require any purification, whereas if you had a direct or indirect technique, you might need more sample prep before you could do your analysis. It's flexible and sensitive because you can have, for your detection, you can use direct or indirect methods. However, it requires some optimization because you have to have, since there are 2 antibodies, you have to make sure that they don't interfere with each other and that they give the highest signal and sensitivity that you can get with that pair. So, with this particular paper, why the authors and why I picked this paper as an interesting topic to look at for bladder cancer is that bladder cancer has the highest reoccurrence rates of all malignancies. So patients, up to even 70% of patients are going to require follow-up screening for quite a few years after treatment. So it's something that you're going to need to have a robust and low-cost and non-invasive technique for your patients to follow through during their course of treatment and monitoring. What's been used in the past has been cytology, so that is really an invasive technique, and also it has a lot of variability between different observers. So ELISA would be a great alternate technique to be used, and what would be ideal would be to find and identify biomarkers that would be useful for screening, and then urine would be a better source for your analysis than blood or serum, because urine is actually a filtrated blood, and it's in direct contact with bladder tissues, so it can actually pick up biomarker proteins that you would not necessarily find in the blood. However, urine is a very complex sample matrix, and it can vary in protein content, concentration, pH, and electrolytes. But, as I mentioned a little earlier in the talk, sandwich ELISA doesn't require the same amount of sample prep and cleanup, so you might have better results using this technique versus your standard direct or indirect ELISA. So, in this paper, the purpose or aim was to develop a multiplex, meaning that several analytes are going to be determined at the same time, and several biomarkers will be determined at the same time to accurately and simultaneously detect, as well as quantify the 10 urinary protein biomarkers for bladder cancer non-invasively. And as I mentioned, bladder cancer is an important area of research, and we're looking to find methods that are going to be less invasive and cost-effective for our patients. So the methods, actually, there's a company called Mesoscale Diagnostics, LLC, and they actually, for this paper, developed a custom-designed sandwich ELISA kit. And I'm not going to list all of these, but there were 10 protein biomarkers that they did screen for. And the sandwich ELISA was a technique used using voided urine specimens. The study had 2 cohorts. It had an initial cohort of 62 that was involved in the initial trial of this sandwich ELISA, and containing 29 patients that had bladder cancer and 33 healthy controls. and this cohort was used to just validate, does the multiplex assay work compared to just the assay of a standard ELISA, looking at each of these 10 individual markers alone. So can we combine this into one assay, was what the cohort 1 study was all about. And then we got the authors got some encouraging data, so they wanted to really further validate it by enlarging the sample set to 200. So in the second cohort, we had 100 bladder cancer patients, 100 healthy controls, and in cohort 2, they just looked at the sandwich ELISA. They didn't look at the individual assay alone. So actually, this group that wrote this paper had done a lot of preliminary work before they even got to this, before they designed this experiment, and optimized it. So at first, there were so many possibilities for biomarkers. So how did they pick the top 10? How did they figure out that these 10 are the ones that we really should look at? So they did a very broad screening of possible biomarkers in bladder cancer from urine in bladder cancer patients using genomics, proteomics, and bioinformatics. And using these broader techniques, they were able to take a very large potential pool of biomarkers and really limit it down to ones that would really be essential and important. So from that initial work that they did, they determined that there were 14 biomarker panels that they need to really further refine to look at. And so from those 14, this group took some commercial ELISA kits from voided urine in cancer patients, taking a sample set of 127 patients, and quite surprisingly, they did a very good job from the proteomics and genomics and bioinformatics to really identify which are going to be the best biomarkers, because 10 of the 14 were actually altered in the cancer patients compared to the healthy controls. So this was really exciting news, and from that, they decided to take these 10 and then further validate it with a sample set of 308 patients. And so they were able to obtain 102 bladder cancer patients and 206 healthy controls. So in this paper, the goal was to find two cohorts. The first one for cohort 1 was to basically see, can we do a multiplex assay? Is it as good as looking at these at doing an assay with each individual target alone? So cohort 1 in this table just shows that you have a variety of different stages of cancer in your sample set represented for both cohorts. So even though sample prep was not completely eliminated, it was pretty simple compared to a lot of other analytical techniques. It just involved a centrifugation, and then the supernatant was going to be used for the ELISA assay itself. So when you're doing ELISA, your samples can have varying amount of proteins. So when you're performing your analysis, you want to control for that. And so one way to do that is to measure your total protein content in your sample. And so there's commercial kits such as Pierce has a kit for normalizing for protein in your sample, so that if you have one sample that has a high level of protein and one that has a low, your results are not going to be confounded by the fact that you have some variation between samples. Also, in order to correct for and normalize for other molecules that are not related to our 10 targets, creatinine was used as a normalization factor. And actually there's another commercial assay that's available for that, an enzymatic assay, and you can read it with a color change readout at a specific wavelength. So here are our individual antibodies that we had, targets that was part of our custom kit. So it's quite a big table here and quite a few different candidates that we were looking at from different vendors. So some of them were matrix metalloproteinases, some are apolipoproteins, some are different enzymes such as carbonic anhydrase, and some are actually growth factors such as the VEGF-A. So this gives you an idea of what was used in the screening for this analysis. So with the actual assay itself, what they did was they took all of these individual monoclonal antibodies from that prior table that I showed you. And so they wanted to make sure that there weren't going to have any negative interactions or suppression or enhancement of signal or anything like that. So they had to sort of do an initial screen before doing the actual experiment to ensure that you would get an unbiased result when they actually looked at the study. So they picked the monoclonal antibody pairs for capture and detection that will yield the highest sensitivity and specificity. And then for detection, they used a monoclonal antibody labeled with ruthenium. And the readout was through chemiluminescence. So enzymatically, a chemiluminescence signal is generated and read on a microplate reader. They performed their analysis using a 7-point calibration curve and had a wide dynamic range of 4 log units. When they were working with the actual samples, they made sure that these urine samples were placed on ice and that they had, with this custom kit, a standard diluent that would reduce what are called matrix effects or differences between your samples, or also with the complex nature of urine to ensure that that's not going to interfere with your assay. So I'm not going to go into all the details here, but you have a certain sample volume, a 2-hour incubation time, a wash, a conjugation with your tagged ruthenium, another incubation and wash, and then you're going to read. So here's the results, and this is actually from the first cohort study where they were comparing, does multiplex ELISA work as well as your individual ELISA? And one index that I didn't learn about until I actually did this project is called this UDIN index. Sometimes it's called the UDIN-J statistic, and it's used in clinical assays to test the performance of the diagnostic test and to see, is this diagnostic test good in terms of adequate sensitivity and specificity. So with this test, if you have a UDIN index that is close to 1, that means that there are no false positives as well as there are no false negatives. So here for both the multiplex assay, we got very high UDIN indexes of 0.9258 as well as with individual 0.9467. So when they looked at statistical comparison of those two, they were not different. So they did perform the same based on the UDIN index. And then the two components of the UDIN index are the overall sensitivity and the overall specificity. And so again, we want both of these values for sensitivity and specificity to be close to 1, because if something is sensitive, then it's going to detect a positive that really is positive, and if it's specific, then it's not going to detect a false negative. So again, we also have 2 other predictive values here. The PPV is your positive predictive value. So basically this is a statistic that determines if you have a positive sample, is it going to really show up positive. And then the negative predictive value is the probability that if you have a negative sample, that it's going to screen negative. And so again, these were close to 1, so overall the assay did perform well. So Here are just the values comparing for the multi-assay on the left and the commercial assay on the right. We have the 10 biomarkers, and we have the mean urinary concentration of them that were assessed by these two techniques. And so the main purpose here was to see, okay, which of these 10 biomarkers are elevated in your bladder cancer patients versus your control. Because obviously if you don't really see a difference, either that means that your assay is not working, or that particular biomarker is not relevant because it's not really showing a difference. And so this p-value is to measure, is there a difference between your bladder cancer sample and your total control. So this p-value is just comparing within the multi-array assay, and then on the right the p-value is comparing within the commercial individual assays. Is there a difference between your healthy control versus your bladder cancer sample? So for the most part, that the study did support and did actually agree with the prior work that these 10 biomarkers are good candidates, and that there is a difference seen between these different techniques. And then when you look at p-values, anything less than 0.05 is considered statistically significant. So for the most part, with a few exceptions, these were statistically significant and showing a difference between your control and your positive bladder cancer sample. So the next thing is, once they looked at the multiplex assay results by themselves and the individual ELISA assay results by themselves, to say, well, do these techniques compare well together? Can we just replace our individual ELISAs with our multiplex? So they did a technique called the area under the curve that they analyzed, and then they looked at this percent difference. They generated a 95% confidence interval, and then they generated p-values. And so p-values, again, anything that is less than 0.05 is statistically significant. So again, in this case, you don't want them to be different, because you want the two assays to compare, because the goal, or the hypothesis is that hey, we could do a multiplex assay and replace the tedious and time-consuming and expensive individual assays. And so for all of these, they were above 0.05. So the group moved on to cohort number 2, and that involved further validation of the multiplex assay. So the goal was to increase the sample size set to 200 total. 100 of cancer patients, 100 of the positive control. And here is just a summary in Table 4 of all of the different biomarkers, and this is for the total cancer and for different stages of cancer, low grade, high grade, and then there are a couple of different special type of cancers, just so you could see just raw values for how these biomarkers are associated with the different cancer types. Okay, and then once they did that, then they looked at some statistics on the method, and like with the cohort 1, they used this area under the curve technique. They calculated 95% confidence intervals, and then they looked at sensitivity, specificity, positive predictive value, and negative predictive value. And so if you get close to 1, or in a percentage 100%, then for these statistical values here, for the 4 that they looked at, then the better the technique. So for the most part, we were getting in the 70s and 80s for sensitivity and specificity. We had a few of these biomarkers that could be improved in the 40s and 50s. But and overall, mostly 70s and 80s for the PPV and NPV percent. So overall, I would say for first pass through that results are really promising for this technique, and that's what the authors felt as their conclusion from this data. So in summary, the authors developed a multiplex assay for 10 biomarkers for bladder cancer in voided urine samples. And this was a custom assay. It had low picograms per ml range limits of detection. So that's how low can you detect your sample. And a linear dynamic range, which means the range of concentrations for your sample detection of 5,000 picograms per ml. And actually, 9 of the 10 biomarkers were actually elevated in your bladder cancer patients versus your control, and there was only 1 that was not elevated. So this work was actually consistent with work that was done earlier with the group that was with immunohistochemistry. So it supports and sort of confirms earlier work also done within the same research group. So from this, it's just really encouraging data that we can use multiplex ELISA, and this was their hypothesis, and so the data supported that. Multiplex ELISA is a promising technique based on this current study for screening and monitoring of bladder cancer patients. It's non-invasive and could be used clinically because it's cost-effective, sensitive, and selective. And then other researchers have reported use of multiplex ELISA, but only in serum, blood serum, and in tissue lysate. So this they've never really looked at urine itself, and so they could be missing out on pertinent biomarkers that actually come into contact with the bladder epithelial cells and those soluble biomarkers. So what they did find is from the biomarkers that were looked at in this screen, there were 2 in particular that appeared to be the most accurate based on the statistics that they looked at, and these were PAI1 and IL8. So the main summary is that results were really encouraging with this, and that further work needs to be done to optimize the assay and to validate it to be used clinically in a doctor or a doctor setting, so that these patients can be evaluated long term. So thank you very much for listening to my whole talk here, and hope that you've enjoyed it. The last 2 slides go over my references that I used for the talk in covering my background. And one thing that I'm going to put a link on with my presentation is an additional MP4 video that is published by this last reference on my list, Thermo Fisher. And I'm just going to give you a link to that, and you can look at that on your own. But it goes over a lot of detail about the differences between direct and indirect and sandwich ELISA. And then today I only really had time to go over the sandwich technique. So thank you again, and I appreciate it.